So they so he started giving the cells out to his colleagues. They were doing some research, but they never got her permission. So she didn't know that they took some of the cells to do research. And then later, what basically using these HeLa cells, they did polio vaccine. They did research for that, and then they used the same cells for HIV, cervical cancer research, HIV identification, and some other research. And total of 60,000 research papers were published, and the research was based on these HeLa cells. And these HeLa cells were from Henrietta Lacks, who didn't even know that they took our cells. So this is more of bioethics thing. So if you're interested, in these topics, maybe take bioethics class. So her family, so after she died, her family didn't know that they had her cells, and then maybe the journalist people, they started going to them and asked how they feel about her cells being alive, and they didn't really understand what they meant. They said, "Oh, the only cells we know is the prison cell. We don't know what you mean. Did they clone her or something?" Because they were not educated enough to understand the science. And guess how much money she got for the research? You guys want to take a guess? Or her family? So yeah, her cells, you can still buy them. None. Yeah, they got zero. I think now they got a couple thousand. Even Oprah Winfrey, she made a movie on it. And if you're interested in that movie, maybe I think it's on HBO Go. You can watch it there if you want to watch that. So this is more of bioethics thing. Even nowadays, if you, let's say, go to Kaiser and you want to get a treatment for something, they're gonna make you sign a bunch of papers. And what those papers are basically saying, anything we take from you, we own it. So that's how they got away with getting sued. And so if you tell them that you don't want to sign these papers, then they will tell you that we can't treat you. So they basically own everything that they take from you. Basically, if they scrap some cells from your skin or anything. So they say that we own that. And later, if they would decide to do some research with it, they can get away with that because you signed that paperwork. So that's bioethics. So Rebecca Skloot, she's the one who wrote about it, and it's called Immortal Life because those cells kept dividing and dividing. And they did a lot of research based on that. Okay, so the definition of a cell, it's the smallest unit of life that can function independently. And we learned about prokaryotes and eukaryotes. And in this chapter, we're going to focus a little bit more on those, and we will learn what's inside of those cells. So here, this is showing you prokaryotic cell. The DNA is floating around. We learned one difference was the nucleus. Here is the eukaryotic cell. It's a lot bigger in size compared to prokaryotes. Therefore, it's going to have a lot more stuff inside. So um, this is the definition. And the word cell, it comes from prison cell. So let me show you something. So if we were in lab, you would have looked at this slide. So this is onion root cell slide. We have these in the lab. And if you look at it, it's boxy. Looks like a prison cell. That's what it would look like. So this is where the term came from. So that's why we call it a cell. So it looks like a cell. It's boxy. Now that's a plant cell. I also want you to keep in mind, later we will discuss how animal cells look different than plant cells. So plant cells are going to have more of a boxy shape, more defined shape. So let's talk about the size a little bit. So students who are international students, maybe who were born in other countries, not USA, maybe they know about the metric system. There is a section in your book goes over the metric system. If you don't understand it very well, please go over that section. So when we talk about size, an ant is about 1 centimeter. And if you don't know what that is, about your pinky nail. That's how big an ant is. Frog eggs, it says 1 millimeter. That's a size of printed period. So if you have a paper, you type a period on it and you print it, that's how big frog eggs are, 1 millimeter. Then you guys have a term that's called micrometers. So this is a letter in Greek, M letter, and then this is English. So this is micrometers. So I might give you a question where I ask you what does this stand for? Then you have to tell me this is micrometers. So 1 micron is 1 meter divided by 1 million. And these eukaryotic cells, the size for these cells is 10 micrometers to 100 micrometers. That's plant cells and animal cells, eukaryotic cells. I might give you a question where I ask you the size of the cell. You will have to remember they are 10 micrometer to 100 micrometer. So there might be a question on the exam about that. And then we move on to bacteria and archaea, archaea. They are 10 to 100 times smaller. So the size for them is 1 micrometer to 10 micrometers. And then under here, you can see it says range of human eye. That means you can see these two with naked eye. For these, you need a light microscope, which we have in the lab, and we would have used them to look at these slides today if we were in lab. And then after bacteria, we have viruses, proteins, atoms, small molecules. These you cannot see using light microscope. For these, you need electron microscopes. So bacteria are 100 nanometers. And nanometers is to the billion. So there's 9 zeros after that. So NM stands for nanometers. This letter here is M in Greek. So this is micrometers. So make sure you guys know those terms. So like I said, you guys need electron microscopes to look at smaller things that are smaller than maybe 1 micrometer. So there are two types of electron microscopes that your book mentions, and it's a transmission electron microscope and a scanning electron microscope. These microscopes shoot electrons, and that's going to create an image for seeing the cell structures. The difference between these two, so before these were as big as size of the room, but as the technology got more advanced, it got smaller in size as well, just like the computers. So the difference between these two, this is a picture showing, this is under transmission electron microscope. If you notice, in this cell, you can see the internal structures in very detail. So you can see them very well. But you can't see what's on the outside too much. Maybe there are some hair-like structures on the outside, but you can't see those. So transmission electron microscope is going to focus on internal cell structures. Let's see. So it's going to focus on internal cell structures, whereas electronic microscope, scanning electron microscope, it's going to reveal details on the cell surface. So if you look at this photo here, you can see on the surface, you can see a lot more detail compared to internal structures. So this is going to focus on the outside of the cell a little bit more. But they're using electrons, they're shooting electrons to create those images because these are too small to see otherwise. This is what we have in the lab, light microscopes. These are used to see bacteria and plant cells and animal cells, so prokaryotes and eukaryotes. They generate color images of living cells. So another thing I want to mention, these colors that you see, those are not the real colors here. So those are just created by the computer because there's no light to see the actual color. So light microscope, you can dye some of the bacteria in pink dye or green dye, and then you can put it under this microscope and you can take a look at it. So you'll be able to see that bacteria and plant cells and animal using light microscope. Light microscopes are less powerful. They don't use any electrons. They use light to look at things. So those are 3 types of microscopes. Are there any questions?
Professor, did you say the size of the cell is, can you repeat that one again? Like 10 to 100 micrometers, is that what you said?
Can you say it again? I couldn't hear you very well.
You mentioned something about the size of a cell.
Yeah,
10
so
to 100
size of
microns?
the eukaryotic cells, plant and animal cells is 10 micrometers to 100 micrometers.
Plant and eukaryote. Okay, got it.
So plant and animal cells, those are eukaryotes. Bacteria is 1 micrometer to 10 micrometers. So there might be one question on that where I ask you what's this cell size, and you're gonna tell me whichever option is true in those multiple choice questions. Okay, so this is one of the big concepts here. One question that I will most likely ask you on the exam is, why are cells so small? So why are they so small? So that's what I'm going to ask you on the exam. This is one of the first concepts we're going to go over, and I told you guys there are about 2 concepts in this chapter. So they're small because smaller cells have more surface area compared to the volume. And if there's more surface area, then it's going to do a better exchange with the environment. It's going to take in food a little bit better. It's going to, maybe carbon dioxide is going to leave and oxygen is coming in. That's exchange, and that's going to be better at it. It's going to be easier to do the exchange if it's higher surface area. So to explain that, let's do some math. I'm not going to give you any math questions on the exam. After I explain this, I will give you an example of the question that you might see. So here, this is a cube here, so 1 centimeter. And then here you have double the size, 2 centimeters. And that's the height and the width, both. And then here, it's a little bit bigger, 3 centimeters, and then 3 centimeters wide as well. And if you are taking geometry right now, or you took geometry, this is the formula for surface area. Maybe you're taking algebra, that's where you have to learn this. So you take the height, that'd be 1 centimeter, width, that's also 1 centimeter, and number of sides. So it has 6 sides. So you have 6 centimeters squared here. You do same thing for the next one, 2 centimeter wide height and width, and number of sides, that's again 6, and that's what you get. You do that for this one, this is what you get. So that's your surface area now. Now we said surface area, smaller cells have more surface area compared to the volume. So now we have to go ahead and calculate the volume. So let's do that. Now the formula for volume is height, width, and length. So height times width times length. So for this one, it's 1 centimeter tall height, 1 centimeter width, and then the length is also 1 centimeter. So you get 1 cubic centimeter here. So 3 is cubic. You do same thing for the next one, 2 times 2 times 2. So height, width, and length, 8 centimeter cubic. And then same thing for this, you get 27 cubic centimeters here. Now what you have to do, we said it's relative, surface area relative to their volume. So we're gonna divide these. So ratio of the surface area to volume. So for this one, it's 6 divided by 1, you get 6. Here you get 24 divided by 8, you get 3. And then 54 divided by 27, you get 2. So you get higher surface area or compared to the volume if you're smaller. And again, that's going to give you better exchange with the environment. So if I ask you this question on the exam, I might give you some numbers where maybe one cell has bigger number for the surface area compared to the other. And I might say which one has better ability to do the exchange with the environment. I'm not gonna have you calculate anything. I might just give you the numbers already. So you're going to answer, cells are small because smaller the cell, the greater the volume to ratio it is, and compared to the larger cell. And the stuff can, carbon dioxide, oxygen, food, waste material can go in and out much easier. So that's